Hi everyone, I'm Jeff. I'm also a postdoc here at CCRC, like Mark and Jatin, who talked before. And I'm going to talk about plants and carbon cycle in climate models, especially land carbon cycle. I'm going to touch a bit on nutrients as well. And this is the outline of my 40 minutes talk, maybe faster. So I give you an overview of the C cycle and some definitions of the acronyms we're using in biogeochemistry. So there's a kind of common way to represent all those processes. So it's the same approach for a lot of carbon fluxes, basically. Then we go a bit more into detail. So how do we simulate the carbon that enters the plant, so the photosynthesis processes? And once it's inside our ecosystem, how is it going to move between a plant down to the litter, down to the soil, and then ultimately released back into the atmosphere? Then something my two colleagues didn't talk about, but I'm going to have a few words about how we evaluate models and the new directions in land biogeochemistry. All right, so this is the carbon cycle. So this is the same picture as Jatin, and that's good because he put the reference. I forgot it. But basically what we want to simulate in the model is how does a plant take up carbon here? Okay, so how does the carbon in the atmosphere enters the leaves? How is it then recycled through the ecosystem or cycle? So this is this turnover process. How does this carbon that's taken by the leaves goes down here? And how is it released back into the atmosphere? Okay, and especially how long does it take? And this is important because plants take up the atmosphere, CO2 from the atmosphere, and release about the same amount. So naturally, the land is kind of neither a sink nor a source of carbon. But if we change the atmospheric concentration of CO2 and also the land temperature, we may expect this will change. And that's why we're modeling that. So the main fluxes are GPP, which is gross primary production. This is a downward flux atmosphere down to the land. And that's a product of photosynthesis. So that's the net photosynthesis. We have RA, autotrophic respiration, which is upward flux of CO2 from the plants into the atmosphere. Autotrophic tells you it's a plant. Plants are autotroph, so they're not like us. They don't need to hunt or eat veggies to grow up. Everything is in the atmosphere already. And heterotrophic respiration is the respirations from the microbes in the soils, basically. That comes from the organic matter decomposition. So when we compute a carbon balance, like in the energy balance, we assign positive values to upward fluxes and negative values to downward fluxes. All right? And then once we know GPP and RA, we get the net primary productivity, which is the net amount of carbon the plant is keeping, and that will become available to be cycled into the ecosystem. And finally, when we add heterotrophic respiration in the balance, we get NEE, which is the net ecosystem exchange. That tells you, and the sign of that tells you if your land surface in a grid cell is a carbon source or a carbon sink. Okay. So now a bit more about biological processes. So I guess you've heard a lot about physics these few days. So the way biogeochemistry presented in climate models is a mix of mechanistic, very described processes, very complicated things. We need a lot of parameters. Some of them we can measure in controlled experiments, etc. But that's not very computationally efficient for climate models. So then we have a mix of empirical models as well. So to go a bit faster in the chains of chemical reactions we're describing. So usually, at least to my knowledge, the way it's done is we define parameters, K, for example, in reference conditions, which is I know the temperature, I know how much water is available, etc., etc. And these are in moles of a product per moles of a substrate per unit of time. You can forget this for now. Parameters vary between plant functional types. So coniferous will not breathe as fast or as much as deciduous plants. This is totally random sentence, but just to let you know. So different plants have different physiology. And then we use response functions to adjust these rates we define for reference conditions. And we adjust the rates based on environmental factors. So this response function will describe how does the temperature affect photosynthesis, how does the water availability will affect the plant growth, how much enzyme availability will affect soil decomposition, for example. And then this process rate, once K has been adjusted by those factors, this is the actual rate we apply to the substrate. So the substrate is the source that is modified by the reaction. So that's an example. Soil organic matter decomposition, D, it's K. We define here reference conditions, say at 15 degrees in optimal water conditions, times a function of temperature in the soil, times a function of soil moisture in the soil, times how much carbon I have in the soil to do the reaction. Okay. A second thing in chemistry that one has to keep in mind is the order of a reaction. So here it was quite easy. So this is the only concentration that's actually altering that result, okay? But in other types, a reaction can be proportional to a substance A, so the concentration of a substance A at a certain power and another substance, etc. So we have partial reaction orders. So here the order for A is alpha and the order for B is beta. And the total order of the reaction R is alpha plus beta. Okay? So the higher the order, the more complicated it is, because there are a lot of things that are going to interact eventually non-linearly with each other. So in modeling, we try to keep things as simple as possible. So most of the time, you will just find first-order kinetics, thankfully, like in my former example. So if you summarize dm equals k times it's actually proportional to the carbon pool size. Okay? Other things, it's a bit of microbiology, etc. But you will encounter terms like Michaelis-Menten kinetics if you read papers. So that basically tells you how much an enzyme catalyzes a reaction. So an enzyme is something that's present in the matter that will link to the substrate that is not modified, but the substrate becomes the product of the reaction. And this is called catalysis. So usually enzymes are not needed to do reactions, but when they're inside the matter, it's really good because it can speed up things by several orders of magnitude. And the way to describe, so I could have put R here, I put V, I don't know why, but basically this is the Michaelis-Menten kinetic equation. So V equals to V max times concentration of S over Km plus concentration of S, the substrate. And Km is one parameter that corresponds to at which concentration my activity V is equal to half my activity V max. So and you can see this is asymptotic reaction. So quite quickly, the reaction will speed up for very low concentration, and at some point it doesn't really matter if we have more available substrate. So that's the way living things work, basically. Okay. The way environmental factors are parameterized, so the effect of temperature, for example, on activity. So by definition, we think that temperature enhances biological activity, which is true up to a certain point. So if the soil is frozen, for example, there's hardly anything going on there. But if it's at good temperature, between 0 to 30 degrees, the biological activity starts up to a certain point. We don't know what will happen if soils go up to 70 degrees or something like this. The change is measured by a value Q10, which is the relative change in activity that follows a 10-degree change in the soil. Okay? So in models, we actually define Q10 ourselves to tell to the model, for example, if I have 10-degree change, so 10-degree increase in activity actually, and Q10 equals 2, then it means my biological activity will double between 10 and 20 degrees. We have other approaches that are called variable Q10 and actually don't rely on Q10 themselves, and this is one example, the Arrhenius function point. And if you plot those in a temperature and value of this thing space, you see that choosing different Q10, so here in blue and red, can give you quite different shapes into the activity. Okay? And if you use these other ones, then you have something that's somewhere in the middle here, but here it's lower, etc. So this is very sensitive in models usually. And you have to define Tref, okay, for which the temperature for which you define your parameter K. Okay. If we speak about water availability, so it's different. So in terms of water, water is at best non-limiting. Okay. Because if there's too much water, usually we have anaerobic condition, which means there's not enough oxygen left for the reactions to go on. So it's as bad as the dryness. And therefore, because it's at best non-limiting, these response functions are much more constrained than temperature response functions. And usually they take value between 0 and 1. So 1 means water is not limiting. 0 means the quantity of water is not good for the activity, whether it's dry or too wet. And this is an example of how it's done in 8 different models of soil decomposition. So each curve tells you as function of ratio of soil moisture to moisture at saturation. So 1 means the soil is full of water, 0 the soil is dry, and this is the response function value between 0 and 1. So some others tells you that below wilting point there's no activity or constantly low. Some others tells you water is good all the way once it passes this point. Other models are quite picky, etc. So the shape of this function is also, the model is going to be very sensitive to the shape of this function as well. Okay, so I'm going to go through photosynthesis. It's going to be a lot of equation in the next few slides, sorry. But it's just to show you how things interact. Okay. So in a leaf, what's going on is this. So we want to simulate this. So that's happening in the leaf. And we want to put that in a climate model with grid cell of several hundreds of kilometers across. So basically, the leaves let air entering, CO2 and O2. And then you have these RUBP, ribulose 1,5 biphosphate, that is catalyzed by this enzyme. And that goes, depending on the availability of those, you go here. So it's Calvin cycle. So this is going to produce biomass. This is the actual photosynthesis. or you go there, this photorespiration. Respiration releases CO2. So the leaf actually does not only take carbon, it also releases some. Okay? There are different pathways of photosynthesis that are named based on how much carbon there's in this first molecule created here. There's the C3 photosynthesis, which is the most common, but it's not very efficient because then the leaf is losing 25% of its photosynthesis by photorespiration. So C3 plants are like normal trees, and why that is maybe it's a kind of evolutionary thing, because when those plants evolved there was much less O2 in the atmosphere. There's the C4 photosynthesis pathway that accounts maybe for 5 to 10% of the whole biomass of the Earth, but 35% of the photosynthesis globally. So what they do, these plants, so they're intelligent, so they trap the enzyme in specific cells, and then they saturate it with CO2. So then it's hardly in contact with O2, and then we maximize the amount of this that's utilized to go there. Okay? And then there's the CAM photosynthesis, which is used by crassulation plants, basically cactus. So what they do is they close their stomachs during the day, because they usually live in places where the evaporation is terrible, and they open it at night to capture CO2. And then they do the photosynthesis during the day when the sun is back. Okay? But basically, all these things are modeled the same way. We just change some parameters in the photosynthesis model. Okay. So now I'm going to go through the common way to do photosynthesis, or at least this is the idea around it. There are a lot of differences between models, but this is the Farquhar's approach from Grant Farquhar from the INU here, actually. And these are two papers that describe the model. This is the original one, and most of the things I've done I kind of copied from that one. So this describes the net assimilation that's happening at the leaf, A. So this is carboxylation, which is when the rubisco is utilizing CO2 and starts to do actually photosynthesis, minus oxygenation, this is the photorespiration, minus RD, this is an ozone respiration. Okay? Those two terms, they depend on the same enzyme, so we can summarize them as the difference between how much the total thing and how much is utilized to carboxylate, because that's what we're interested in. Okay? And this gamma star is the photosynthetic CO2 point. I don't go too much into details here, because you see that in current models these things are set fixed or parameterized in much easier way. So then, but if you go in a really mechanistic way, you have to define this, the ratio of this on CO2 concentration is equal to that. So it's how much the rubisco prefers CO2 to O2, and then you define this, and you find back this Michaelis constant from earlier, etc. Just going quick here. And then once you have this term here, okay, you need to know VC, and VC is calculated as the most limiting factor. So we compute the carboxylation 3 times, and we choose the lower value we achieve. So the thinking is, if I need water and temperature, then the thing that limits me the most, it's the actual thing happening. Okay? So this is rubisco limited carboxylation rate, so how much enzyme do I have in my leaf, RUBP, so how much RUBP I have in my leaf, etc. TPU is this trisphosphate. So all those things are some steps into this Calvin cycle I showed before, where you calculate them. So these are horrible equations, of course, to show on a slide. So, but it always basically depends on oxygen and carbon dioxide concentration. And here you have a term J, sorry. That's electron transport rate that you can calculate with that. Okay, so wow, very complicated, but thankfully, since it was originally published in 1980, it's been modified and adapted dozens of times. So we use gamma as a parameter, so we avoid a lot of complicated equations. Simpler parameterization of J as function of J max, so we don't have to have this PPFD alpha, I don't know this, and beta. So we have like 4 parameters that we have to define. No, 5 actually, you have that one here. That's very complicated, okay? So based on observation, we find simpler ways to parameterize J as function of temperature again, and a maximum value we can achieve. There's been different ways to calculate temperature effect on temperatures, as I've touched on a bit earlier. And now these things are coupled to stomatal conductance models. It will tell you how much stomats open during the day, so how much transpiration can happen. Okay. And in climate, so actually the model I've just described, it's very detailed. Okay, so you could use it at the tree scale. But in climate models, we apply big leaf models. So basically, we think a tree is just 2 leaves or 3 leaves that will have different position in the canopy. So some are sunlit, top of the canopy, some are shaded at the bottom of the canopy. And actually using big leaf models or very complicated different layers of leaves, it doesn't really matter. And at the end, simulations are very comparable in quality. Just that if you have 2 leaves to calculate all these things, it's much faster, it's much more efficient. Okay, so now I'm going to talk how this carbon that enters the leaves, how it goes down in the tree and the soils, etc. So first, photosynthesis, GPP, half of it is re-released by the plant into the atmosphere by autotrophic respiration. And the autotrophic respiration, RA, is the sum of the maintenance respiration, which means how much energy the plant is using to stay alive, and growth respiration, which is how much the plant is using to build up new biomass. So how much a tree uses to go up. Oh, that was written here. And good. And so as I said before, NPP, which is the difference between photosynthesis and respiration, that's the actual amount of carbon that remains available in the ecosystem. So the maintenance respiration is usually simulated as first order reaction. So this is quite similar to the decomposition equation I showed before. So we have respiration here. It's a function of a rate that we input in known conditions times C above C2N ratio in the leaves times a function of temperature. C above C2N ratio is actually just how much nitrogen I have in my plant. So this can be fixed in some models if you don't do nitrogen yourself. This is fixed, and then we have once again this function of temperature to describe biological activity. But this is fairly simple then. Just have to know the shape of the Q10 thing. And the other part of respiration, it's a fraction of the difference between GPP and maintenance. So it's not really calculated in most of the model. So so now we got NPP. Where was it? Okay, yeah. So that's what enters the tree. So it has to be allocated to different plant components. So and plants are usually constituted of 3 pools. This is a claim from me, because that's how it's done in the model I'm using. But basically you can imagine we have leaves, we have wood, the trunks, and what's under the ground, so the roots. So from the earlier step, we calculated the autotrophic respiration for each pool. We sum it up, and we remove it from GPP, and we get NPP. And then NPP is divided between all the pools instantaneously. Okay. And sorry. Okay, it's between all three pools, and based on when we are in the season, there's going to be more or less going to the leaves, more or less going to the wood, more or less going to the roots. So this can be either fixed or calculated dynamically depending on how much the tree needs. Okay. So once the carbon is in my plant, how is it going to be recycled by the litter and soil? So basically, as you all know, plant parts die, so the leaves fall, the branches break if it's windy, some roots die because it's too dry and they become useless, etc., etc. So the way to simulate this is to say there's a turnover rate or residence time of carbon into each of these pools. And this is once again calculated assuming first order kinetics once again. So D for dying plant is one parameter, which is the residence time, so how much the carbon would stay, say, in my leaves if the temperature was optimal or equal to 10 degrees, for example, times a function of temperature, a function of water availability, times how much carbon I have. And this will give me the amount of carbon that's actually leaving the leaves every time step. This term will change between different compartments. So carbon will stay longer in the wood than in the leaf, usually. Okay? Then once this flux is leaving the pool and is then reallocated to a different pools. And here, litter pools. So here I have my trees. So 3 boxes, basically. So I know the carbon in the leaf is going to leave down to go in a metabolic and a structural pool. From the roots, the same, but wood. So falling branches, basically, they go in what's called coarse woody debris pool. So the litter to keep in mind is if you go undercover in a forest, that's all the leaves in the ground. And if you actually dig into that, you have the first layer is the new leaves from the last year, and then if you go a bit down, you have leaves, they're already a bit altered, already a bit decomposed, etc., etc., until you reach the soil. So it's a kind of pre-soil environment. Okay? So the carbon in the litter pools is decomposed by heterotrophs, so microbes. And in each litter pool, there's a specific turnover rate adjusted by environmental factors applied to the pool, assuming first order kinetics. So the decomposition by microbes in the litter pools is basically the same formulation as before, just that you replace K by its value for the litter pool, and Cl is the amount of carbon in the litter pool. Okay, and if you do the balance, so what enters the litter pool is what came from the plant P minus what's leaving it. So parts of this decomposition is allocated to soil pools, microbial, so and passive in that case. And part of it is released as CO2, so back in the atmosphere. That's part of autotrophic respiration flux. And once we go in soil pools, so we're going to start having residence times or values of K of the order of centuries or millennia. In litter, we were speaking in terms of years. Now we're going to speak in terms of centuries, decades. And then, okay, so once again, carbon in soil pool is decomposed by heterotrophs. And I've already seen that. In each soil pool, there's a specific turnover rate adjusted by environmental factors, assuming first order kinetics. So once again, decomposition in the soil is the rate adjusted by environmental factors times how much I have to decompose. This is the balance. So what enters in the soil is the decomposition from the litter minus what was released by respiration from the litter minus what's decomposed here. Can be exchanges between soil carbon pools. So the microbe is going to take up some from the slow carbon pools, etc. And basically this K, which is the residence time, it tells you how hard it is to utilize these molecules for living things. It's a measure of lability. But so once again, decomposition is allocated to other pools and released as heterotrophic respiration. Usually it's a fixed ratio. Okay? And then we sum up heterotrophic respiration for litter, soil pools, etc., etc. And then we have NE, which is NPP we had from all the plant calculation, and heterotrophic respiration. So this is mission accomplished. And but the story is not over for our little friend. So now we've modeled the things we wanted to simulate, but of course we have to evaluate how good our models are. So the processes I've been talking about are very detailed, okay? So, and usually these are 3 scales things we're describing. So we need very high resolution measurements. But the monitored areas are few, okay? So you're not going to find measurements well distributed across the planet. So we also need global kind of synthetic data sets to compare our models when we apply it at global scale. So for a site scale, which is, so usually when we use those models at the site scale, is we isolate one part, which one grid cell of the model, basically. And we just run the model for one grid cell. And to the best data set that exists to match models with observation is the FluxNet network, which is distributed here. So it's a network of eddy covariance towers. So all these triangles represent one station, and the color is the different networks. So there's a North American one, a European one, an Australian one, etc. So you see there's one problem that would first arise from these data sets. Although it's very good with long-term high-frequency stations, etc., there's a huge bias toward Western Europe and the United States. So basically, we have no idea what's going on here. I mean, this is Sahara, so maybe not much in terms of carbon exchange. I mean, no offense. But here, say in the Congo region, which is very green area, it will be very interesting to monitor what's going on here in terms of forest, etc. So at the global scale, thankfully we have satellite data, so MODIS products, etc. But we also have synthesized data, so which are kind of empirically upscale from field observations. So the Oak Ridge National Laboratory data sets, the harmonized soil database, and this is a soil database just for high latitudes in the northern hemisphere, etc. But all these are usually fluxes measurements. How much goes in, how much goes out of the soil. But in all the equations I've been talking about, there's always the store, so how much there actually is. But this is not usually in data sets. And this figure shows you the state of the art of measuring fluxes. So this is the accuracy and the precision of measurements. So the bottom left corner is high accuracy and high precision. And we see that basically just GPP and NPP are very well known in terms of measurements and how good we can measure it. But respiration fluxes, which are the two things that are going up in the atmosphere, it's already not very well known, and we cannot really measure it because we cannot measure the soil everywhere, and the soil is very heterogeneous, etc., etc. Yeah, so new direction about biogeochemical modeling. So I've just been talking about carbon, but we know that carbon, nitrogen, and phosphorus cycles are tightly linked, that plants need nitrogen and phosphorus to grow. That's why we expand fertilizer on fields, etc., etc. And it's actually a kind of hard relationship because plants will need nitrogen from the soils because they cannot take up all the nitrogen that's in the atmosphere, which is too bad because it's about 80% of the volume of the atmosphere. But they need it from the soils to build up the leaves that use them to take up carbon, okay? But at the same time, they need carbon from the atmosphere to build up the roots that allow them to take up nitrogen. So this is a very tough a balance between processes, roots and leaves processes. And in this paper by Jeff Exbrayat, this just shows you an evaluation of how much nutrient availability limits plant production in the world. So the more red it is, the more we would benefit to put fertilizer on those areas to improve how much plants are growing. And so you see here in western South America or in middle China. So these are very red areas, but here as well we have about 50% decrease in growth just because there's not enough nutrients. And whereas here in Europe, where we've been fertilizing for centuries now, the limitations are very low because all what we put on the field stay for some time as well in the soil and become available through decades afterwards. We try to improve new modules. So how does forest fire? How much a forest fire would release carbon in the atmosphere, which is very important for the Australian carbon balance, for example. How do wetlands and peatlands evolve through times?